Hello my student, I'm your biology teacher, Chip Govin. Our topic today is chapter 11, Inheritance, and our subtopic is 11.4, Inheritance in Humans. Here are the learning standard. Learning standard number 1, identify type of human chromosome, autosome and sex chromosome. Number 2, analyze and describe human karyotypes. Number 3, match paternal and maternal chromosome of human in a drawing or micrograph to build a complete karyotype of an individual. 4. Relate Mendel law to human inheritance. Number 5. Analyze human inheritance by constructing inheritance cross diagram. And number 6. Analyze human inheritance by studying family pedigree. Inheritance in humans. There are two types of human chromosome, namely autosome and sex chromosome. Human somatic cell consists of 44 autosomes and 2 sex chromosomes. Here are the comparisons between autosome and sex chromosomes. Based on feature, autosome consists of chromosome pair from number 1 to 22, whereas sex chromosome consists of one chromosome pair that is number 23. Based on functions, autosome control all characteristics of somatic cell, whereas sex chromosome consists of gene which determine gender of the individuals. Example of autosome is type of blood group, height and skin color. And for sex chromosome, male has XY chromosome, whereas female has XX chromosome. Autosome vary in terms of size and length. Male sex chromosome of XY are different in size. X chromosome is longer than Y chromosome. Y chromosome only carry genes which determine sex characteristics. In this slide, there are two types of karyotype, the male karyotype with sex chromosome XY and female karyotype with sex chromosomes XX. The number and structure of chromosome present in the cell nucleus is known as karyotype. So this is male karyotype with 44 plus XY, and this is female karyotype with 44 plus XX. Chromosomes are arranged in pair based on homologous chromosome in terms of their size, centromere locations, and bending patterns of chromosomes. So pair number 1 until pair number 22 we know is called autosomes. Okay, chromosomes are arranged in number by size, starting from the largest pair to the smallest pair is autosomes, and the pair number 23 is the sex chromosomes. So this is the male and this is females. Non-disjunction is changes in number of chromosomes occur due to failure of homologous chromosome to separate during anaphase 1 or failure of sister chromatid to separate during anaphase 2. So the male gamete or female gamete can possess chromosome number of less than 23 or more than 23. So here there are three types of non-disjunctions: Down syndrome, Turner syndrome, and Klinefelter syndrome. Down syndrome have 47 number of chromosomes, so extra chromosome for chromosome pair 21 here, so also known as trisomy 21. Down syndrome can occur in both male and female. So these examples are karyotype of male with Down syndromes, okay, the XY here. And this one is Turner syndromes. The gender of individual with Turner syndrome is a female. The total number of chromosome is 45. There is a missing of X chromosome in a pair of sex chromosome here. And Klinefelter syndrome has 47 number of chromosomes. There are extra X in a pair of sex chromosome here. So Klinefelter syndrome is a male and his secondary sex characteristics are not well developed. ABO blood groups. ABO blood group is in multiple alleles. Blood group is controlled by three different alleles, namely alleles IA, IB, and IO. However, a person only possesses two alleles to determine his or her blood groups. Both allele IA and IB are dominant allele, whereas IO is a recessive allele. Individual with blood group A can be either homozygous dominant A or heterozygous dominant A with recessive allele O. Okay, so blood group B can be homozygous dominant IB or heterozygous dominant with one recessive allele IO. Individual with group AB both are dominant allele, allele IA and allele IB. So allele IA and IB show codominance. This means that when both alleles are present, they both have an effect. Individual with group O, there is no dominant allele, both are homozygous recessive allele present. So this is the blood group O. Okay, this diagram shows the blood group ABO systems. IA represent antigen A and IB represent antigen B. So individual with group A have antigen A, individual with group B have antigen B, and individual with blood group AB have antigen A and antigen B, and blood group O, there is no antigen present on the red blood cell. So you have learned about the ABO blood group when you were in Form 4. Okay, look at these questions. A man with A blood group married a woman with B blood groups. Explain the probability of the couple in getting a child with O blood groups. So how to get O blood group? O is a recessive trait. So to get the recessive trait, this means the couple must have the recessive alleles. Okay, so this is the genetic diagrams. So the parents is heterozygous A blood groups and the mother have heterozygous B blood groups. So this is the meiosis process and this is the gamete IA, IO, IB and IO. So this is fertilization process when allele with IA cross with the alleles with IB. So this one is AB blood groups and this one is heterozygous A, one is recessive alleles O. So this one also this one is A blood group, this one is B with one recessive allele O. So this one is B blood group, and this one is the only carry the both are recessive allele. So this is blood group O. So the phenotype of child is AB, A, B and O. So the phenotype ratio is 1, 1, 1 and 1. So answer, both mother and father are heterozygous A and B or the blood groups to get a child with the O blood groups. Rhesus factor. Beside antigen A and antigen B on the surface of human red blood cell, there is another antigen called antigen D, which is known as rhesus factor. An individual whose red blood cell has rhesus factor is said to be rhesus positive, whereas individual without the rhesus factor is said to be rhesus negative. Okay, we look at this situation, situation number 1 and situation number 2. Situation number 1, the father and the mother both are homozygous. The father is homozygous positive, rhesus positive. The mother is homozygous rhesus negative. Situation number 2, the father is heterozygous rhesus positive. The mother is homozygous rhesus negative. Okay, both are the same. So homozygous rhesus negative. This is heterozygous rhesus positive because positive is dominance over rhesus negative. Okay, this is the gamete. Okay, this is the gamete. Inheritance of rhesus factor from parents to children is based on principle of Mendel's law. Rhesus factor is controlled by genes which consist of pair of alleles, namely rhesus positive dominance and rhesus negative recessive. Genotype of rhesus positive individual is either homozygous dominance or heterozygous rhesus positive. Rhesus negative individual is homozygous recessive. So both are Rh negative, Rh negative. This is homozygous recessive. This is heterozygous because one is positive, one is negative. This is homozygous dominance because both are rhesus positive and rhesus positive. Look at these genetic diagrams. Situation number 1, When the father is homozygous rhesus positive, crossed with the mother who is homozygous rhesus negative, we can see the genotype of child is all are heterozygous rhesus positive. Okay, RH positive, RH negative. You can see all is RH positive and RH negative. So this is heterozygous dominant rhesus positive. Okay, so phenotype of child is rhesus positive. Okay, look at situation number 2. When the father is heterozygous RH positive and the mother is homozygous rhesus negative, you can see the genotype of children is, this one is homozygous rhesus negative, this one is heterozygous rhesus positive. Okay, there is dominant alleles and recessive alleles, dominant allele and recessive allele. This one is both are recessive alleles. Okay, so this is rhesus positive, this is rhesus negative. So the ratio is 1:1. Okay. Thalassemia. Thalassemia is an inherited disease. The disease can be passed down from generations to generations. Thalassemia is due to gene mutation on an autosomes, that is on chromosome 11 or 16. Thalassemia is due to the abnormality and low number of hemoglobins. The red blood cell is smaller and paler. Look at this genetic diagram. Situation number 1, mother is normal and the father is carrier. So this is the gamete. Both are normals, but this one carry one recessive alleles for thalassemia. And when these normal alleles cross with normal allele from father, the children is normals. This one is carrier because when we cross the normal allele with the recessive alleles, the children is become carrier, and this one is normals, and this one is carrier. Thalassemia carrier is said to have a thalassemia minor. This means individual does not show any symptom of the disease, just it carry the recessive allele for thalassemia. Situation number 2, mother is carrier and the father also carrier. So this is the gamete. Okay, one of the gamete carry one recessive allele. So when we cross the normal allele with normal alleles, the children is normal. But when we cross the normal allele with the recessive alleles, the children is carrier, and this one also carrier. But when both alleles are recessive, cross with the recessive alleles, so the children is become thalassemia. Okay. Sex determinations. A male has 44 plus XY chromosomes and a female has 44 plus XX chromosome. So altogether is 46 number of chromosome. So when during meiosis process, the number of chromosome reduced into half. So in males, they have two type of gamete, 22 plus X and 22 plus Y, whereas female only one types of gametes, 22 plus X and 22 plus X. When fertilization occur, so this is the products. So this is 44 plus XX, this is female, and this one is 44 plus XY, this is male, and this is male. So the phenotype ratio is 1:1. Sex-linked inheritance. Genes located on sex chromosome which control specific characteristics but are not involved in sex determinations are known as sex-linked genes. Characteristics of color blindness and hemophilia are caused by recessive genes linked to X chromosome. Y chromosome is shorter than X chromosome and does not contain as many alleles as X chromosome. Therefore, in male, any trait caused by a dominance or recessive allele that is found on the X chromosome will be expressed. Look at this table for color blindness, the phenotype and the genotypes. Normal phenotype for male, there is a dominant allele at X chromosome, while there is no allele at Y chromosome. Normal phenotype for male, there is a dominant allele at both chromosomes X. Carrier, male cannot become carrier because male have only one X chromosome, so if X in male carry recessive allele, the male become color blindness and not become carrier. Female can become carrier when one of the chromosome carry one recessive alleles. Color blinds for male, when one recessive allele presents, the male become color blindness, but for female, there needs two recessive allele on both X chromosome for the female become color blindness. Color blindness is a condition in which a person cannot differentiate some specific color, such as red and green. Color blindness is caused by recessive allele found in the X chromosome, and most people with color blindness are male, because male wants recessive allele at X chromosome enough for the male become color blindness. Okay, look at this one, the table for hemophilia, the phenotype and the genotype. The normal males with dominant allele at X chromosome and there is no allele at Y chromosomes. Normal female, both X chromosome have dominant alleles. Okay, male cannot become carrier. Female is carrier when carry one recessive allele at X chromosomes. Hemophilic male when one X chromosome carry recessive alleles. And hemophilia female when both X chromosome carry recessive alleles. Hemophilia is a condition in which blood cannot clot in normal circumstances due to lack of blood clotting factor. This can result in excessive internal or external bleedings, which may be fatal. Look at this genetic diagram, a cross between father and mother with normal eyesight. Mother is carrier because carry one recessive alleles. So when meiosis process occur, the father produce two types of gamete, X chromosome which carry dominant alleles and Y chromosome does not carry any alleles. So mother produce two gamete with X chromosome, one carry dominant alleles and one carry recessive allele. When fertilization or process occur, this is the child genotypes. This is girl because XX, so both are dominant alleles, so the girl is normal. This one, the girls carry one recessive allele, so the girl is normal, but we call it carrier because carry one recessive alleles. This is boy because XY, so this one is dominant alleles carried by X chromosomes, but this one is color blindness because the X carry the recessive alleles and Y does not carry any alleles, so the boy with color blindness. So the phenotype ratio is 1 normal girls, 1 normal girl with carrier, 1 boy normals, and 1 boy with color blindness. This is genetic diagram for hemophilia. Okay, look at the parent phenotype. The father is normal males and the mother is normal female, but the mother is carrier because carry one recessive allele at X chromosome, and when meiosis process occur, the father produce two types of chromosomes, chromosome X and chromosome Y. Chromosome X carry one dominant allele, but chromosome Y does not carry any alleles, and the mother carry chromosome X with dominant allele and chromosome X with recessive alleles. So when fertilization process occur, so this is the genotype of childs, 2 girl and 2 boy. This girl is normal, but this one is carrier because this carry one recessive alleles. This one, the boy is normal, and this one is hemophilics because carry one recessive alleles at X chromosome. For the boy, one recessive allele enough for the boy becomes hemophilia. Okay, so this is the phenotype ratio. One is girl normal, one girl normal with carrier, one boy normal, one boy hemophilia. Ability to roll tongue and type of earlobes. The ability to roll tongue and type of earlobes are two characteristics that can be inherited from parents to children according to Mendel law. Ability to roll tongues is a dominant trait. Free earlobe is a dominant trait, whereas attached earlobe is a recessive trait. So unable to roll tongue also a recessive trait. Family pedigree. Family pedigree or lineage can be analyzed to investigate inheritance of human characteristics. Family pedigree is a flowchart through a few generations to show ancestral relationship and inheritance of characteristics from ancestor to individual in the present generations. So this diagram shows the hemophilic inheritance in a family, and this is the generation number 1, generation number 2, and generation number 3. And this is the symbol used in pedigree. Square shape is a normal male, round shape is a normal female, square shape with color is a hemophilic male, round shape with a color is hemophilic female, and round shape with half color is a female carrier. Okay, a first generation marriage will produce the second generations. Okay, the first generation we can see the couple, one of the male is normal and the female is hemophilics. So the second generation we can see the male both are the hemophilics and the female is a carrier. Okay, so when the male with hemophilic married with a female with also hemophilics, so the result is all the children in third generations or are hemophilics. And this one, carrier, female married with the normal males, so one of the children, which is the male, is hemophilics and the other children is normals. So you can see that the male is, the probability to have hemophilic is higher compared to the females. Formative practice 11.4. Question number 1. A woman has heterozygous B blood group and a man has heterozygous A blood groups. What are the chances of children having A blood group? Explain the inheritance of blood groups of this family by using a schematic diagram. So this is the schematic diagrams. So the father and the mother, and this is the genotype, heterozygous A blood group, and the mother is heterozygous B blood groups, and this is the gametes. Okay, so this is the progeny genotypes. This one is carry allele IA and IB, so this is phenotype AB blood groups. This one carry one dominant allele IA and one recessive allele IO, so this blood group is A. This one dominant allele B and one recessive allele O, so the blood group is B. And this one carry both recessive allele IO or allele recessive O, so the phenotype of progeny is O. So the ratio is 1 AB, 1 A, 1 B, and 1 O. So the probability of having A blood group is 25% or 0.25. Question number 2. A woman who is heterozygous for hemophilia married a normal man. What are the chances of the couple having a hemophilic children? So we have to draw a genetic diagram. Father is normal. Mother who is heterozygous for hemophilia. So the mother must be carrier because one of the chromosomes carry recessive alleles, one carry dominant alleles. Okay, so for color blindness and hemophilia, Okay, both are sex-linked inheritance, so X and Y chromosome must be shown when writing the genotypes. Okay, this is the gamete, fertilization process, genotype of child. This one is female, this one is females, this is normal, this is carrier, this is normal males, this is hemophilic males, so girl normal but carrier, boy normal and boy hemophilics. So the phenotype ratio and the probability to get hemophilic children is 25% or 0.25. Question number 3. A man who is homozygous for the ability to roll his tongue married a woman who is heterozygous for the ability to roll her tongue. Predict the phenotype of the offsprings. So this is the genetic diagrams. Man is homozygous, woman is heterozygous. So the parental genotype, this one is homozygous dominant, this is heterozygous dominance. Lysis process, gametes, and the genotype of filial 1 generations. This is homozygous dominance, heterozygous dominance, homozygous dominant, heterozygous dominant. So this is the phenotype of filial 1 generations. All can roll tongues. So this is the filial 1 genotype ratio, 1 to 1s, and the phenotype ratio is all are able to roll her tongues. So the children can roll tongue, 50% are homozygous, whereas 50% are heterozygous. That's all for today. See you in next lessons. Thank you and bye-bye.